Very good morning, students. In this lecture, we'll be talking about the intracellular transport in Paramecium chordatum. In the previous lecture, we had discussed about the amoeba protease, that how the transport it occurs in amoeba. So in continuation with the previous lecture, now this is the second part of that particular lecture. This is the intracellular transport in Paramecium. So as you all know that Paramecium chordatum is a ciliate and also belongs to the phylum Protozoa. It lives in the fresh water, so that's why it has got the same problem that the amoeba had of osmoregulation. So what happens that 24 hours it is under stress of the incoming water due to endoosmosis. Suppose this is the Paramecium and it is living in fresh water. So what will happen, this water is in low concentration of salts and the Paramecium has got a high concentration of salts. So what happens with osmosis? This water will move inside the body of the Paramecium. So this is endoosmosis, as we have discussed in amoeba. So this intake of water, it also serves for the intracellular transport in Paramecium. Now what happens that Paramecium has got two contractile vacuoles. First vacuole is over here and the second contractile vacuole is here. Now these contractile vacuoles, they have got fixed position. This is very important, that they have got fixed position, and secondly they have the fixed number. And next, one thing is fixed more, it is the outpour of the contractile vacuoles. So these are the characteristics of the contractile vacuoles which are present in the Paramecium. So as it is living in fresh water and this water is coming inside the body of the amoeba, the Paramecium. Now what happens, these contractile vacuoles, they are taking up that extra water. So in amoeba, we had discussed that amoeba had got a large number of contractile vacuoles, and the number of the vacuoles, that was dependent upon the quantity of the water that is coming inside the body of the organism. So here also, whenever the fresh water it enters the body of the Paramecium, the two contractile vacuoles, with the help of their radial canals, now these are the radial canals. So we make these contractile vacuoles like this. So why like this? Because these are the radial canals of the contractile vacuoles, and these radial canals, they are helping in gathering of water. So the water is gathered into the contractile vacuoles like this. So they take up water from all the sides, and the whole of the water, it reaches the center of the vacuole. So that is how it takes up the water, extra water inside it. So once the water it enters the contractile vacuole, it bulges out. So when the contractile vacuole it bulges to its full, then it contracts. See the lower contractile vacuole. So what happens is that it will contract to a very small size. It will contract. So when it will contract, it will give out whole of the water from the dorsal side. So I have told you that contractile vacuoles, they have got fixed outpores, and these pores are at the dorsal side, so they are not at the ventral side. So now we are seeing the ventral side of the paramecium. So the water will leave the body from the side which is away from us. So whole of the water from the body of the paramecium, it will go out. Now what happens is that when the water was coming inside the body of the amoeba or the paramecium, it is coming in. Are you seeing this? Okay, you just take this red arrow. The water is coming in. It is going inside the contractile vacuole. Then the contractile vacuole, it bulges up, and then this water is removed from the body of the paramecium through the outpore of this contractile vacuole. Whole of this cycle, it results into a phenomenon which is called as cyclosis. So this is a very, very important feature in case of paramecium. So this cyclosis, it helps in the movement of the food particles along with the other structures inside the body of the paramecium. So that was the first factor that helped in the cyclosis movement or cyclotic movement inside the body of the paramecium. So that was the entry of the fresh water. So that was due to endosmosis. So that is again an intracellular kind of transport. Now, so if I take the first factor into account, this is the endosmosis. Now if I take the second factor into account, the second one, I take it as this will be indigestion, or I should write it as like movement or intracellular transport due to indigestion. So here what happens that the paramecium has got a particular oral groove over here. So if I draw it, this is the oral groove, and so we clear it. This is the oral groove, and it has got a number of cilia over it, and these cilia, when they move, they are forming a water current, and this particular row of cilia is called as the endoral membrane. So this endoral membrane, it is helping in the formation of water current. Now when these cilia they move, what happens that a water current is formed, and that current is coming towards the inside. So the direction of the movement of the water is towards the mouth or this particular oral groove. Now what happens that if we take up the oral apparatus of Paramecium, if I complete it, it is just like this. So I should make it with the black pen. It is just like this. At the very tip here, this is the mouth actually. So this is the mouth and this is the oral groove. So this part is the oral groove, and below is present the mouth. So whenever the water current is coming inside this oral groove, it goes down up to the mouth. So along this path, the food also enters into it. Suppose this is the food. These are the food particles. Blue ones, these are the food particles. They are coming inside the oral groove. The small food particles, they are given through the mouth, and a membrane is pinched off just like this, and food particles they enter into that particular membraned part, and this pinches off. It pinches off with the food particles. Now this is what we call as the food vacuole. So this is the formation of food vacuole inside Paramecium. Now this blue colored body is the food vacuole, and it is containing the food. Now with the cyclosis, this particular food vacuole, it moves inside the body of the Paramecium. Now how it moves is a typical path. It goes up, takes a turn, and comes back, and again it takes a turn, and that takes a shape of 8. In the shape of 8. So this is a very peculiar 8-shaped path of the food vacuoles, and ultimately when the food is digested, the mechanism is the very same. I am not going to repeat the mechanism. That lysosomes, they attach to the food vacuole, the enzymes of the lysosomes, they help to digest the food, then the food is taken into the cytoplasm, absorption occurs, then the whole of the absorbed food is taken out of the food vacuole, only the residual food is left inside the vacuole, and that is called as the residual body. Now this residual body, it goes to a particular place in the Paramecium which is called as the cytopyge. Cytopyge. So this is the particular place which it comes to at the very end when it has evolved into a residual body. So this particular part is called as the cytopage. Cytopage is also referred to as the anus of the paramecium. So this particular region is called as cytopage. So food vacuole, after moving in the shape of 8 many times in the paramecium, ultimately it goes to the cytopage. Now here the outermost membrane of the paramecium, which is called as the pellicle, is very thin. So this particular membrane, it breaks off, and from the cytopage, whole of the material or the residual material, it is thrown out of the body of the paramecium. So that is a kind of the mechanism of ingestion, digestion, absorption and egestion in case of paramecium. Now how it happens? It is due to the cyclosis. Now here the movement of number third factor can be, now here I should give it number 3. So these are the movements in the oral apparatus. So the movements in oral apparatus, they also help in the formation of cyclotic movements inside the paramecium. So what we are studying is actually that how intracellular transport it occurs in the body of the paramecium. So now we are discussing the factors which are making the cyclotic movements. So here I can give another factor, number 4. It will be the movement of endoral membrane. This is again one of the crucial major factors that are producing the cyclotic movements inside the body of the paramecium. So the 5th factor we can attribute it to the contractions in the contractile vacuoles. So these are the 5 factors which are helping in the formation of the cyclotic movements or the intracellular transport movements inside the body of the paramecium. So hope that you will you have understood this particular topic. So here your heading should not be digestion, or your heading should not be ingestion or the outtake of the food, but here you should make headings like this. So that was about the paramecium. Thank you.